Good evening. My name is Walter Park, and I have the privilege of being the immediate past president of the Collaborative Alliance for Pancreatic Education and Research, and I'd like to welcome you to our first in a series of virtual Pancreas Academies. I want to thank you all for taking time out of your busy schedule in participating. We think this will be an educational endeavor over 5 series in adapting to the COVID pandemic. Historically, Pancreas Academy was a one-day in-person event meant to provide kind of primary core material regarding pancreatic diseases. And over the next 5 weeks, we will host a series of 1-hour sessions of 2 thought leaders. So the format of the webinar, this will be a 1-hour session. I'm just going to briefly go over some housekeeping and introduction. The lecture will be 20 minutes each with a 10-minute Q&A per speaker at the end of the session. As this is a webinar, I presume many of you have become used to virtual seminars and webinars. We will have you on mute as well as your video off. This will be disabled. Questions, clarifications, technical comments, please use your chat function. And this is how we will, this is how I, as the moderator, will see your questions and prioritize them for the guest speakers. If you'd like to change your username under the participant chat list, this is how you can do this. You could click on your name and then rename it if you wish. This activity has been planned and implemented in accordance with the accreditation requirements. It is a CME, it's 5.0 AMA credits. New this year, there's MOC and CEU credit designation. Hopefully, most of you will have had an opportunity to complete the online pretest. The questions were developed by the speakers and they correspond to these presentations. And following the meeting, we will send all attendees a link to an identical post-test to see if the collective knowledge of the covered topics improved. Individual scores are only accessible by the survey administrator and will not be shared. Attendees who complete both the pre and post-test will be entered into a drawing to win a $100 gift card. Here is the link to the website as well as a QR. To claim the CME and MOC, you must complete the following after each webinar, and there's 5 webinars, so you must do that today, both the evaluation form and the post-test. This will be sent to you by email. Your program evaluation form and request for your CME certificate will be processed by our CME provider, and this will take approximately 4 to 6 weeks. This program would not be possible without the support of educational grants, and we'd like to thank the following companies for their grant support this year of this academy: AbbVie, AstraZeneca, Medtronic, and Olympus. Furthermore, we would like to thank our corporate sponsors, and we would encourage all attendees to visit our sponsorship page where it houses a virtual exhibit hall. If you click onto this link, which is the sponsor link, you can check in and be entered into our drawing for a $50 gift card, and 2 prizes will be randomly awarded amongst those who check in on the exhibit site. This is an optional activity separate from our CME program. I just want to highlight that next week we will have Dr. Chris Forsmark and Dr. Mark Lowe. The following week we'll have Dr. Nathan Bahari and Dr. Amy Lucas. August 13th, we'll have Dr. Tamal Turks and Dr. Shonak Majumdar. And concluding on August 20th, we'll have Dr. Anna Phillips and Dr. Catherine Morgan. So again, I want to thank you for attending. Please feel free to follow us on Twitter at Caper Pancreas, or subscribe to our YouTube channel, or follow us on Facebook. At this point, I'm going to turn the screen over to Dr. Aatur Singhi, who will lead off on our first lecture of this academy, focusing on the function anatomy of the pancreas. Dr. Singhi is an associate professor of pathology at the University of Pittsburgh. Dr. Singhi.
Thank you, Walter. There we go. So, sorry, just going to minimize everything. And so thank you, Walter, again, and thank you, Caper, for the invitation to speak to everyone here today that logged in tonight or today, depending on what time zone you are. For those that don't know me, as Walter mentioned, my name is Aatur Singhi. I'm an anatomic pathologist at the University of Pittsburgh Medical Center with a subspecialty training in pancreatobiliary as well as molecular pathology. And today I'll be providing a primer, so to speak, of the pancreas that includes the anatomy, physiology, function, and a little of the pathology along the way to serve as a basis for the remaining talks within this series. Now, in the brief amount of time I have, there are a few objectives I'd like to cover. First, it's best to start with pancreatic development, which will give everyone an idea of how congenital anomalies can occur. Next, I'd like to give an overview of essential pancreatic anatomy that will nicely segue to discussion of pancreatic cellular constituents and basic functions and digestion. And finally, in discussing various components of the pancreas, it's inevitable that we will touch upon some aspects of pancreatic pathology. So let's start in the beginning. The human pancreas develops during the 4th to 7th weeks of intrauterine life. It is the result of 2 outpouchings of endoderm from the primitive duodenum at the junction of the foregut and midgut. These 2 outpouchings are referred to as the dorsal bud and the ventral bud. Within the next few diagrams, the dorsal bud is designated in black and the ventral bud is designated in red. And as I hope everyone will appreciate, that the union of the dorsal and the ventral buds as they form the pancreas proper is truly an intricate process that I equate to an extremely well-rehearsed dance. As organogenesis starts, the small ventral bud rotates behind the duodenum dorsally from the right to left and fuses with the dorsal bud. The ventral pancreatic bud develops into a portion of the pancreatic head, while the dorsal bud forms the majority of the pancreas to include the pancreatic neck, body, and tail. In addition, as these buds fuse, so do their ducts into a common channel that becomes what is known as the main pancreatic duct or duct of Wirsong. In addition, the proximal portion of the dorsal bud forms a small accessory duct or duct of Santorini. As the pancreatic buds and ducts merge, the main pancreatic duct also merges with the common bile duct that drains the gallbladder and liver. Both the pancreatic and common bile ducts merge juxtaposed to the duodenum at the level of the ampulla of Vater. The ampulla is simply a sphincter that controls the release of both pancreatic juice and bile into the duodenum. Once this process or dance is complete, it results in the formation of a single gland known as the pancreas. But again, this is a very complex process, and as one could imagine, things can go awry, such that if something or someone crashes the party, or that complex rotation of the ventral bud as it fuses with the dorsal bud fails, and multiple congenital anomalies can occur as a result. For example, rather than fusion of the dorsal and ventral pancreatic buds, very often both ducts remain distinct and separate. This is referred to as pancreatic divisum and occurs in approximately 10% of the population. Pancreatic divisum is the most common congenital anomaly of the human pancreas. The most common form of pancreatic divisum is classic pancreatic divisum, where the ventral duct continues to merge with the common bile duct at the ampulla of Vater, but becomes strictured over time, while the dorsal duct or accessory duct of Santorini remains patent and dominant, functioning as the main pancreatic duct as seen here. Another congenital anomaly that can result due to malrotation of the pancreatic ducts, albeit rare, is the annular pancreas. This condition occurs due to the growth of a bifid ventral pancreatic duct that encircles the duodenum upon fusion with the dorsal bud. In other words, the duodenum becomes surrounded by the entire pancreatic gland, and interestingly, if a brisk amount of acute or chronic inflammation occurs in the annular pancreas, this can result in duodenal obstruction. Now, at the time of birth, the newborn pancreas weighs a mere 2 to 3 grams and rapidly increases in weight by adulthood to 80 to 120 grams. But the weight of the gland gradually decreases after the age of 40 years, and by the age of 90, weighs around 70 grams or less. Anatomically, the pancreas is situated in the retroperitoneum at the level of the 2nd and 3rd lumbar vertebra, just right of the midline, and stretches to the left laterally across the posterior abdominal wall toward the hilum of the spleen. Now, unlike other organs in the retroperitoneum, the pancreas is not an encapsulated organ. It does not have a membrane that covers it, but rather blends almost imperceptibly with the associated retroperitoneal adipose tissue. It's nestled within the semicircular shaped second and third portions of the duodenum. Anterior to the pancreas is the left lobe of the liver, and as the gland tapers to the left laterally, it lies underneath the gastric antrum and superior to the proximal jejunum. Grossly, the normal pancreas is pink, tan to yellow, and uniformly lobulated. It's composed of 5 rather indistinct anatomic regions: the head, uncinate, neck, body, and tail. The vascular sulcus, or vascular groove, is very helpful in orienting the pancreas as it runs across the neck of the pancreas. The vessels present within the vascular groove include the portal vein, which drains both the superior mesenteric vein from below and the splenic vein left laterally. Sectioning of the pancreas reveals white to tan ducts extending into the well-demarcated lobules of the pancreas. The main pancreatic duct, or duct of Wirsung, averages about 3 millimeters in diameter. Tributaries to the main pancreatic duct consist of up to 50 interlobular or branch ducts, and really an innumerable number of intralobular ducts that drain into those interlobular ducts. Microscopically, the pancreas is arranged in about 1 to 10 millimeter lobules. The pancreatic parenchyma within the lobules consists almost entirely of epithelium, with minimal intralobular connective tissue and adipose tissue separating the lobules. The ducts are often found within the center of the lobules and surrounded by variable amounts of fibrosis. A simplistic way of understanding the pancreatic ductal system is to equate it into basically a tree, with the main pancreatic duct represented by the trunk, the interlobular ducts as the branches, the intralobular ducts as twigs, and finally the leaves along the branches represented as the pancreatic lobules. The basic functional unit of the pancreatic lobule consists of the Acinar Parenchyma, which makes up approximately 85% of the pancreas. The intercalated duct, which is the smallest duct outside the acini, drains the acinar secretions and merges into the intralobular ducts. And lastly, the islets of Langerhans. The islets serve as the endocrine component of the pancreas and constitutes about 1 to 2% of the gland volume in adults. Interestingly, it consists of about 10% of the gland volume in newborns. In addition, although the islets are distributed throughout the pancreas, they're actually somewhat more prominent or numerous in the tail of the gland. The exocrine portion of the pancreas is largely comprised of the Acinar Parenchyma that synthesizes, stores, and secretes digestive enzymes such as protease, lipase, amylase, and others. In addition, the exocrine pancreas also includes the ductal system that secretes bicarbonate and water from the intercalated ductal epithelium. Together, the digestive enzymes, bicarbonate, and water constitute what is known as pancreatic juice, which you'll hear a little more about in the next couple sessions, which is then secreted into the pancreatic ductal system that eventually leads to the main pancreatic duct, then the ampulla of Vater, and finally into the duodenum. Focusing on the acinar cells themselves, they're pyramidal-shaped cells and are ultrastructurally composed of basally oriented nuclei, rough endoplasmic reticulum, and numerous apical located zymogenic granules. The zymogenic granules contain 3 major groups of enzymes that are critical to digestion. The pancreatic protease enzymes, trypsinogen, chymotrypsinogen, that are inactive forms of trypsin and chymotrypsin, amylase that hydrolyzes the carbohydrate known as starch, and then lipase that hydrolyzes triglycerides, as well as other proteases and pancreatic enzymes. These proenzymes and enzymes are synthesized and secreted from the Acinar Parenchyma. Subsequently, they're expelled into the ductal system. In addition to the Acinar Parenchyma, the intercalated pancreatic duct also contributes to the formation of pancreatic juice. Pancreatic duct is equipped with numerous ion transporters that deliver a large amount of bicarbonate and water to the apical surface of the ductal epithelium, which is also secreted into the pancreatic ductal system. However, the net amount of enzymes, bicarbonate, and water secreted by the exocrine pancreas is actually relatively small. The pancreas needs really help pushing those enzymes out, and thus the majority of pancreatic secretion is due to duodenal stimulation through hormones, CCK and secretin. CCK is released by specialized duodenal enterochromaffin-like cells called I cells, and is in response to the presence of lipid in the duodenum. CCK primarily stimulates the release of acinar enzymes. Secretin is also released by the duodenum, but from the S cells, and is in response to acid in the duodenum. In turn, secretin stimulates the secretion of bicarbonate. Once in the duodenum, enteropeptidase, also called enterokinase, that is present on the duodenal surface activates the proenzymes, trypsinogen, chymotrypsinogen, and procarboxypeptidase. All three proenzymes undergo proteolytic cleavage, resulting in their activation, and their active forms result in autocleavage of their proenzymes, potentiating a cascade of enzymatic activity, just a positive feedback loop. While these enzymes are necessary by digestion, one can also appreciate how their dysregulation can be responsible for a number of pathologic entities, one of which is acute pancreatitis. Acute pancreatitis, to at least the pathologist, is defined as a reversible disease process characterized by acute inflammation of the pancreas. There are several causes and possible mechanisms, but all roads lead to autoinjury, meaning where the intercellular enzymes auto-digest the pancreas, leading to injury and an inflammatory process. Acute pancreatitis has fairly specific macroscopic and microscopic findings. In mild cases of acute pancreatitis, the lobular appearance of the pancreas is not as defined, and there is intralobular edema. Further, there is both fat and parenchymal necrosis, as well as saponification. As acute pancreatitis becomes more severe, so does the intralobular edema, fat, and parenchymal necrosis with saponification. This is followed by peripheral hemorrhage around the areas of necrosis as nearby vessels are injured. Severe cases of acute pancreatitis or necrotizing pancreatitis feature extensive fat and parenchymal necrosis with saponification of the pancreas and peripheral hemorrhage. Microscopically, parenchymal necrosis is characterized by destruction of the normal acinar parenchyma, which is the most labile portion of the pancreas, as compared to the ducts and islets of Langerhans, and replaced with fibrin, where only cellular outlines remain of the acinar parenchyma, or really a shadow of once was there that's left behind. Histologically, fat necrosis is characterized by loss of the normal adipocyte cell membranes with fibrin deposition and basophilic casts due to lipolytic enzyme digestion. In contrast to the reversibility of acute pancreatitis, chronic pancreatitis is a progressive condition characterized by irreversible damage to the exocrine and endocrine components of the pancreas. The etiology of chronic pancreatitis can be similar to acute pancreatitis, but also includes genetic and immunologic factors. For example, hereditary pancreatitis is characterized or associated, with mutations in the genes PRSS1 and CFTR. Macroscopically, the features of chronic pancreatitis include overall atrophy of the pancreas and intralobular fibrosis with accentuation of the remaining pancreatic globules. Additionally, there's duct obstruction, which often results in interlobular ductal dilatation. Long-standing chronic pancreatitis can lead to pseudocyst formation, which is basically non-epithelialized cysts filled with necrotic debris. Microscopically, chronic pancreatitis is characterized by extensive loss of exocrine parenchyma, which again consists of acinar and ductal epithelium. There's also prominent thick fibrosis and dilatation of the residual ducts. Residual islands of acinar parenchyma are often surrounded by thick bands of intralobular fibrosis. Now, as opposed to loss of the acinar and ductal epithelium, the islets of Langerhans are relatively spared, but also surrounded by that thick fibrosis, literally islands amongst the sea of fibrosis. Interesting, genetic forms of chronic pancreatitis, like hereditary pancreatitis, are not associated with broad areas of fibrosis, but rather extensive adipose tissue or fat. Further, these patients, as well as others with chronic pancreatitis, often harbor multiple precancerous lesions called pancreatic intraepithelial neoplasia, or PANN. PANNs are microscopic neoplasms that arise from the intralobular and intercalated ducts of the pancreas and are precursors to pancreatic ductal adenocarcinoma, and seem to undergo a sequence of genetic and histologic changes on their way to pancreatic cancer. However, PANNs are very common in the general population. In fact, by the age of 60 years, almost everyone has a low-grade PANN, and therefore only a subset will progress to pancreatic cancer. Histologically, low-grade PANNs are characterized by apical mucin production, and as these PANNs start to accumulate genetic alterations, they become more papillary in appearance, and then the nuclei become elongated. Eventually, low-grade PANNs become more hyperchromatic and architecturally complex, and finally, high-grade PANNs have marked nuclear and morphologic abnormalities similar to adenocarcinoma, but has not become an invasive process. Now, it's estimated that 85% of pancreatic cancers arise from pannins, which begs the question, what's the origin of the remaining 15%? Well, the remaining 15% of pancreatic cancers arise from what is known as mucinous pancreatic cysts that include intraductal papillary mucinous neoplasms, or IPMNs, and mucinous cystic neoplasms, or MCNs. As opposed to pannins, which are microscopic lesions, both IPMNs and MCNs are macroscopic lesions. And as by their name, IPMNs are really characterized by this papillary, basic florid appearance, while MCNs are extremely mucinous and characterized by distinctly cellular stroma. But more importantly, IPMNs and MCNs can be identified by standard radiographic imaging, such as CT and MRI, and thus, considering they are precursor neoplasms that give rise to 15% of pancreatic cancers, and visually apparent, they are in theory amenable to early detection strategies. However, once again, a precancerous neoplasm that is broken through the basement membrane of the duct are then classified as an invasive process or a pancreatic ductal adenocarcinoma. Macroscopically, pancreatic adenocarcinomas often arise in the head of the pancreas and can be found centered around the pancreatic duct. The arrow here points to the cancer, which, to be honest, can be actually very difficult to identify for even the pathologist, and often seen obliterating normal structures surrounding the pancreatic parenchyma. In fact, one can see how normal lobular architecture of the pancreas is literally lost and destroyed by the adenocarcinoma. Moving along and trying to, I'm sorry, staying a little on time, I'm a little off, but in addition to the acinar parenchyma, the pancreas also includes, in addition to the acinar and ductal parenchyma, the pancreas also includes the islets of Langerhans, which are responsible for the endocrine function of the pancreas. The average human pancreas contains approximately 1 million islets. Although morphologically uniform and monotonous in their population, the islets actually consist of at least 4 distinct cell types and supporting vasculature. The 4 cell types are alpha cells, beta cells, delta cells, and PP or gamma cells. By far, the majority of islets are composed of beta cells. The alpha, beta, and delta cells all secrete hormones. Alpha cells produce glucagon, while beta cells produce insulin, proinsulin, C-peptide, and amylin, and delta cells produce somatostatin. All 3 of these cell types are involved in overall glucose homeostasis. In contrast, the function of pancreatic polypeptide that is secreted by PP cells is not entirely known. However, it has been shown to decrease contraction of the gallbladder and inhibit pancreatic exocrine function. Similar to the ductal epithelium, neoplasms can also arise from the islets of Langerhans and are typically composed of 1 or even 2 cell types. These neoplasms often start out as precancerous neoplasms, just like pannins, and are known as pancreatic neuroendocrine microadenomas. Microadenomas are defined as less than 0.5 centimeter and represent a clonal outgrowth from a single islet of Langerhans. In this case, an alpha cell that is producing glucagon that started to proliferate into a microadenoma, which contrasts a normal islet, as seen by the red arrow, that's expressing mixed expression of both glucagon and insulin. Once these microadenomas are larger than 0.5 centimeter, boom, they're designated as a pancreatic neuroendocrine tumor. Pancreatic neuroendocrine tumors can occur throughout the pancreas. Macroscopically, they can vary significantly, including broad variability in size, color, consistency, but in general, they're well-circumscribed lesions. Microscopically, pancreatic neuroendocrine tumors are often walled off or encapsulated from the uninvolved parenchyma by a band of fibrosis, but can be arranged in multiple organoid patterns that include nests, trabeculae, as well as sclerotic forms, as seen at the bottom right. However, despite the variety of patterns, they all have a common cytologic finding, which is eosinophilic granular cytoplasm and stippled or salt and pepper nuclei. They ideally all express neuroendocrine markers that include synaptophysin and chromogranin A. And so I know my time is kind of fleeting, and I just wanted to wrap this up by just giving you a couple of take-home points as you go through the rest of the series of the CAPER talks. Just to keep in mind as you walk through all of these lectures, but to really review some of the pathology that I've talked about, or at least the histology. First, the pancreatic gland develops through a complex dance of 2 buds, the ventral and dorsal bud. Unfortunately, some dancers don't make good partners, which in case of the pancreas can lead to a number of congenital anomalies like pancreatic divisum. Further, the pancreas can be viewed as having 2 main functions, the exocrine and the endocrine components. The endocrine function is performed by the islets of Langerhans that contains 4 distinct cell types, 3 of which are involved in glucose homeostasis. In contrast, the exocrine function of the pancreas is performed by 2 cell types, the acinar parenchyma and ductal epithelium, that are responsible for producing pancreatic juice. If the exocrine portion of the pancreas is perturbed, which can occur due to a number of etiologies, such as acute pancreatitis, the acinar parenchyma is very labile and results in tissue destruction by its own digestive enzymes. If that injury becomes irreversible, chronic pancreatitis occurs and is often characterized by sweeping fibrosis, at least in the sporadic cases. Unfortunately, chronic injury to the pancreas can lead to neoplasia with the development of precancerous lesions called pancreatic intraepithelial neoplasia, or PANIN, which can progress from low-grade PANIN to high-grade PANIN and ultimately pancreatic ductal adenocarcinoma. But PANINs are microscopic precursor neoplasms. There are two others that I want to make sure everyone remembers, which are macroscopic precursors that can be picked up by radiographic imaging. Specifically, IPMNs and MCNs are intraductal papillary mucinous neoplasms and mucinous cystic neoplasms. Finally, it's important to remember that pancreatic ductal adenocarcinoma is not the only form of pancreatic cancer. Pancreatic neuroendocrine tumors, or PANITs, are the second most common malignant neoplasm of the pancreas, and similarly arise from a precursor neoplasm known as pancreatic neuroendocrine microadenomas. And that concludes my whirlwind tour of the pancreas. I hope everyone is still with me. I know it's a lot of information, and therefore, if you do have any questions, I'm happy to answer them. If not, feel free to send me an email later if you'd like. Thank you.